Kerry, can you share the slides? Good morning, everyone. Welcome to our technical panel on COVID-19 in low resource settings. I'm really delighted today that we're going to be focusing on what we're calling testing news you can use. We have a great lineup of experts here to join us today. I'll start with those who are listed on the slide. We have Dr. Melis Anahtar. She's a fellow at Mass General Hospital. Dr. Tyler Miller, also a fellow at Mass General Hospital in Pathology. We have Dr. Stanley Juin, who's with the Center for Disease Control and Prevention in Haiti. He's also joined by Melis, who's a clinical lab specialist with CDC Haiti. And then for Discussants, we have Dr. Eric Rosenberg, who's the director of microbiology lab at Mass General Hospital and a professor at Harvard Medical School. We have John Iafrate, who will also join us, also a professor at Harvard Medical School. Dr. Vanessa Kerry, who's the founder and CEO of Seed Global Health, and I'll be moderating today. So we have some formal presentations, and then we'll also be taking your questions for the panelists to answer. If you have questions, please use the Q&A feature in the Zoom webinar, and I'll be moderating those. So thanks to all our panelists for joining us and you in the audience. I'm going to pass first to Dr. Stanley Juin in Haiti. Welcome, Stanley. Go ahead.
Okay, thank you, Louise. I think you did a good introduction for us, so we don't need to go over that. So let me quickly say what I'm going to talk about. I'm going to talk about the challenges that we are facing improving lab capacity in Haiti, mainly in the age of COVID-19. And next slide. Okay, so what do we have on the market in terms of testing availability for COVID testing? So we have multiple test kits that have been developed to rapidly detect protein from COVID-19. And we also have rapid diagnostic based on antigen detection and on antibody detection. And we have routine confirmation by PCR and serologic method on the way, like ELISA based on detection on IgM and IgG antibodies. Next. Next, okay. Now, there are also point of care testing available for COVID-19. So some are based on antigen detection and others RDTs on antibody detection. So the antigen detection uses the level of viral protein. So you have high level of concentration is required to be positive. False positive category from rapid diagnostic tests are likely. They are also based on antigen detection. So the antigen needs to be present to be detected. And it's also tributary of the presence of early infection. So the typical influenza RDT specificity range between 30 and 80%. I'm not sure we have that specific numbers for RDTs for COVID available now. For antibodies production are late in the infection. So based on what we find, antibody detection can be detected around 9 to 10 days. So roughly 2 weeks after the start of the disease. So you have a high possibility of false negatives and also false positives. Next. Can you close? Yeah. So here in the country, in terms of capacity for testing, currently there are 2 central labs habilitated that can perform the test for the country, and both of them are using PCR, and both of them are using PCR, And with the Abbott, we are capable of doing about 200 to 400 tests a day. And with the ABI platform, we can also test for an additional 270 per day. So we are very far from capacity, even though we are planning on ramping up the testing. But as of now, since we have tested roughly about 450 or close to 500 tests, so we are very far from capacity. There is also the possibility of decentralization of testing using the gene expert. As you probably know, probably Louise more than you guys, but we have about 40 or 46 gene experts on multiple areas in the country. So we have the capacity to use this platform on all 10 geographic departments of the country. So at the central lab capacity, we are next. At the central lab capacity, we are using currently the molecular methods. And routine confirmation of COVID cases is based on the detection of the RNA of the virus by real-time PCR. Several molecular diagnostic protocols are available, but multiple assays are also available, both that can be used both on Abbott and ABI. And molecular testing is still very expensive, and there is high degree of resistance to rapid testing here in country. Next, in terms of COVID challenges, there's difficulty validating multiple assays as many countries faces global shortage in terms of reagent. Here in country, we don't produce them, so we have to procure them on open market. And there's a big competition for lab reagents, and we have a shortage on everything, and we need to go through PAHO to get everything we need, since we cannot really compete with other countries. And as you know, the US, they block shipping of anything COVID related, including PPEs to other countries. So we cannot really find anything but going to PAHO. So there's also an increased need of staff, since now we are planning on ramping up testing, and we want to do the testing on multiple points throughout the country. So there's a need of trained personnel to perform those testing, trained in terms of infection prevention and control, and also in terms of specimen collection, specimen transport, everything. So we do have a plan to decentralize. I think I've talked about that already. But we also don't have a site-specific risk assessment. And before doing it, it would be ideal that we could do this site-specific assessment, so we could plan, target more where we're going to do the testing, where they are ready to do the testing. And they also must be equipped in terms of infrastructure, such as either certified type A1 or A2 biosafety cabinet. Requirement to have also an adequate plan to manage biohazard waste. The transport system, we think that our transport system currently will quickly be overwhelmed, since it wasn't designed to bring sample every day. The plan was for the system to bring our sample twice a week, but with COVID, samples are being transported every day to the national lab. So we are at a breaking point, and with this ramping up of testing, we feel like the system will quickly be overwhelmed. There's also the feedback issue. The national lab was not designed to send feedback quickly to physicians. So whenever you have the test, and we are dealing with a great issue here now, because physicians, they expect the result to come quickly. And even though we test, our turnaround time for COVID is 24 hours, which is good. But the test, it takes time for the test to get to the department or to the physicians because the result, because the lab was not, they typically send result back to the department and not on a routine basis. So basically on a monthly basis, they can send a report on what they have found, how many positive, how many negative by category, but they are not set up to send the result to physician unique for individuals. And for COVID, they need to do it. So it's really a struggle to send feedback to the field. There's also with multiple labs, needs for infection prevention and control grows exponentially. And with this global shortage of almost everything needed to care for COVID, we really are dealing with issues of shortage for everything. So with this ramping up of testing, we think it's going to be worse and we will probably need to adjust. One last thing I need to talk about is the use of the rapid test. And it's more the IgG/IgM rapid test. Currently in country, they don't really want to use them because I think maybe some of you know, some partners have some, they brought them and they are using them at their facility or at the border. Using them for public health purpose is not bad, but using them for clinical diagnostic is prohibited now in the country, although we know some partners are still using it. It's a high sensitivity subject in country because the national lab doesn't want to use them at all. But for surveillance, the ELR would like to use them in some setting to do some type of screening, like on the borders. So there are ongoing discussions related to the use of the rapid test, but as of now, they are not accepted by the Ministry of Health. So I think I tried to cover quickly what I could in terms of challenges for the lab, and me and Manis were hoping for your questions. Thank you.
Thanks a lot, Dr. Stanley, Juin, and Manis. I think we probably have a lot of questions, and I'll come back to ask you some of those. But I think in particular, you highlighted a few issues, which is I know because we've known each other a long time, and I know Manis, you have a lot of technical expertise in Haiti. And as often is the case, it sounds like your capacity at the moment is really limited by the lack of materials. You don't have reagents and you don't have PPE to be able to do the testing and the equipment. So let's come back to that as well. I know that in the next presentations, we'll also talk a bit about what we're calling rapid diagnostic tests, which are a lot the antibody or maybe some antigen tests. So we'll come back to discuss that with you. Thanks very much for your presentation. And we'll move on to Dr. Anahtar.
Good morning, everyone. So my name is Melis. Thank you for the introduction. I'm the microbiology fellow at Mass General. Next slide, please. So Dr. Juin has already given a beautiful introduction to this already, but I'll just briefly describe the two main testing methods that we are using for COVID-19, which are PCR and serologic testing. And the optimal test really depends, of course, on the clinical scenario. So Tyler Miller will speak next about serologic testing, so I'm just going to focus on the role of PCR. So PCR is, for us, the gold standard of diagnosing COVID-19 within symptomatic patients. And as Dr. Juin said, it's really most sensitive during the first week of illness, as the viral loads really peak, it looks like around 4 days after the onset of symptoms. So at MGH, in addition to using it for diagnosis, we also use it for deciding when patients can be taken off of precautions, and in particular, when healthcare workers who have confirmed COVID-19 illness can return to work. They need 2 negative tests spaced apart by 24 hours or more. For epidemiologic purposes, since viral shedding really only occurs within the first several weeks, serologic testing is a much better test for epi. Next slide, please. So real-time PCR is certainly not a new technology. We've been using it routinely for many other viral load assays that we do in our hospital, like CMV and HSV, HIV viral loads, etc. But for a really quick refresher, if it's been a while for anyone, real-time PCR really builds upon the basic principles of PCR, but it kind of cracks open that black box and lets you monitor the amplification reaction as it's occurring in real time. So in addition to your forward and your reverse primer, you have an addition of a TACMAN probe that binds in between your viral gene of interest. And as the polymerase is zipping along that template strand, it cleaves the TACMAN probe, releases fluorescence, and that fluorescence signal increases with every PCR cycle as your template is being exponentially amplified. If you could click, please, to show. And so that's monitored in real time, and you get these nice curves. And the point at which the fluorescence signal rises above a certain threshold is called the cycle threshold. And the earlier the cycle threshold, the more virus is in your original sample. Next slide, please. So the reason real-time PCR has become the gold standard for symptomatic patients is that it has some very desirable characteristics. It's very specific in that you need 3 SARS-CoV-2 specific binding events to occur. You need your forward primer, reverse primer, and your probe. And in addition, most of these real-time PCR assays have 2 targets, so you can definitely trust that a positive result indicates that this is SARS-CoV-2 infection. And the assays are generally highly sensitive as well, where assays can pick up as little as 1 genome copy per microliter of input. And it's semi-quantitative, where your CT value roughly is proportional to the inverse of your viral load. So if your patient is showing up with CT values in the teens, you can extrapolate that they have quite a high viral load in their nose. Next slide, please. And there are now an increasing number of qPCR assays that are available. It's important to note that they actually have significant differences, even amongst the 30 or so that are now FDA approved. Many of them require special instrumentation, like a specific brand of a qPCR machine or an existing instrument platform, like the Abbott ID Now device. Some of them require QIAGEN extraction kits, which can be really hard to come by these days. Some of the tests are approved just for NP swabs. Others you can use on sputum and BAL. And there's also marked differences in that limit of detection, anywhere from 0.04 to 100 copies per microliter. And that really affects your overall sensitivity of the assay. And interestingly, the assays also assay for different parts of the viral genome. Next slide, please. So at MGH, over time, we've really undergone an evolution of our PCR testing strategies. We started with a qPCR lab developed test that we adapted based on the CDC assay to the instrumentation that we have in-house, which was not the EUA approved applied biosystems instrument, because we didn't have that and it was back ordered for a month. So at the time in early March, since we didn't have any commercially available assays, this was our only option. And I'm happy to just have offline discussions of how we set up this assay if anyone's interested, but it is extremely laborious. It requires manual RNA extraction, manual setup of the qPCR 96-well plates. It's very error prone, but it works. And even though it could only run 20 to 30 tests per day at the time, that was still very useful for us. The samples did have to be batched, so the turnaround time suffered as a result. So because our hospital had a really high test volume requirement, we moved to the Roche Cobas 6800 instrument as soon as it became FDA EUA approved. This is a massive instrument the size of a car. It normally runs all of our HIV, hepatitis, and CMV viral loads. And when we repurposed it for COVID-19 testing, we were now able to get through 300 to 400 tests per day on that instrument. And it eliminates a lot of the human error that's associated with the lab developed test. And since everything from sample extraction to the qPCR is all automated. And because it is batched in batches of 94 samples, the turnaround time varies anywhere between 4 to 16 hours, depending on when we get that run in. So there are definitely situations in our hospital that need faster turnaround time, particularly within the ED or within obstetrics. And in those patients, we test using the Cepheid Gene Expert System, which I imagine many of you are routinely using. It sounds like in Haiti you'll be using as well. We really like this test. It's extremely convenient. It has a very fast turnaround time of 45 minutes. it has an amazingly low limit of detection of 0.25 copies per microliter. It's actually our lowest limit of detection of the assays that we have in-house. The limiting factor for us is that the cartridges are still unfortunately in limited supply. We're only allocated to get 40 tests per day. So we found it to be very accurate. It can run 24/7. We would run all of our tests on it if we could. We have the Infinity instrument for running the Cepheid in. And what's not shown on the slide is some other assays that we're working on that are up and coming that would be useful in lower resource settings. Like we are working on a technology called LAMP, which works within about 30 minutes directly from the sample. It doesn't have the same sensitivity as PCR. It's only about 70% sensitive, but it will hopefully be a nice rule-in assay. And then there are also CRISPR-based lateral flow assays that other people are working on as well. Next slide, please. So even as clinical pathologists, we're asked a lot about the sensitivity of qPCR and whether there are false negatives. And it's really hard to actually pin down that number of what the sensitivity of qPCR is. And I'm sure you've heard that Chinese studies that have stated sensitivities in 70% range. Of the 1,000 or so patients that we've tested multiple times at MGH, we've only found that 1% had an initial true false negative. And we found that these false negatives tend to happen for 3 reasons. The first is obvious. If there's poor sampling, then we're not going to be able to pick up the virus. The second is if the patient is tested in the pre-symptomatic period, then they develop symptoms several days later, and the PCR is positive when they're actually symptomatic, but wasn't at that initial time of testing. And the third situation that we see pretty commonly is that patients have been sick for more than a week. They're presenting now with predominantly lower respiratory tract infections. And we know from the published data that's shown on the right here that the sensitivity of nasopharyngeal PCR decreases later on in infection, while the sputum PCR can remain positive. So in some of these patients, we pick up the infection by sputum PCR. But it's also possible that in these patients who've been sick for a long time, serologies may be positive. So I'll now turn it over to Tyler Miller. He's going to speak about serologic testing that we're doing here, but happy to take any burning questions if there's any.
Thanks very much, Melis. Why don't we, I'll ask the audience, please don't be shy to ask questions in the Q&A. It's okay if they're like basic questions about testing. I normally hand the specimen at the lab and I just wait for the results. And so please don't feel shy to ask those questions and we can come back to them and we'll go ahead and have Dr. Miller give his presentation as well.
All right, everyone. Thanks for the introduction. So I'm going to talk about serological testing. And so you've heard about the PCR and antigen testing. And I think the first question is why would we do serological assays for COVID-19 and what were we looking for? And so on the left here on your screen is sort of a typical immune response to viral infection. And so you have sort of this incubation period where you have the initial infection and then some period of time before they get symptoms. And during that period, your body can sort of sense the virus, but hasn't necessarily mounted an immune response yet or become symptomatic. And then for most people, sometime after they become symptomatic, they start to produce an IgM antibody response, and that's followed by an IgG antibody response. And so you can detect these using a number of different assays that I'll go over today, one being an ELISA and 2 being sort of a point of care lateral flow assay. Now the question becomes, why would you use these assays? And I think the most obvious answer is to look at the prevalence of sort of past and present SARS-CoV-2 infections in a population. As Melis said, the point of, or the PCR assay is great for the acute infection, but afterwards it's hard to tell if somebody had been infected because they're likely no longer to have viral titers. And so there's, identifying the infections in a population and then identifying potentially individuals with immunity, right? I think the jury is still out on if that's the case for COVID-19, but that's certainly a promise of this test. And if that's the case, then you can figure out ways to identify those individuals with immunity and come up with protocols for return to work. And It's also possible in large-scale screening efforts that you would identify asymptomatic patients that are still potentially infected or infectious. People who have like an IgM response are potentially still infectious. And then the state of a disease response within individuals, and then also identifying high titer donors for therapeutic plasma donation. Next slide. All right, so how does this work? So I'm going to basically focus on the lateral flow assays and the point of care testing, which is what we have validated in-house now and are using. I also think there's a lot of utility for these in low resource settings. And so this really only requires a finger prick of blood, and then you take that drop of blood and put it onto this lateral flow cassette, add a little buffer, and within 10 minutes you have an answer whether somebody has IgM or IgG or both antibodies. Next. And the way this works is that you essentially add the patient's blood to a sample pad that will then contain the antibodies. This flows over a conjugation pad here. This conjugation pad has a COVID-19 antigen, which acts as the bait that binds the antibody. This also contains a gold nanoparticle, and then this flows over this nitrocellulose membrane. And if you have a human IgG or an IgM, there are strips of anti-human IgG or IgM on this nitrocellulose membrane that bind those specific isotypes and will become visible with that gold nanoparticle. And then, the readout is essentially like a pregnancy test where you have a control line, which is at the top to show that the assay is valid. And then you have an IgM, IgG, or both. Next. So at Mass General Hospital, we have brought in 3 different brands of these and validate them in our sort of validation cohorts. The biomenomics test is manufactured in China, but is being sold in the United States through the Biomenomics company and Henry Schein. And then SC Biosensor is a South Korean company, and Biolytics is a Singapore company that's being also manufactured in China. Next. So for our validation, we had essentially used 60 negative samples that were all collected pre-COVID-19 outbreak. There were 30 asymptomatic patients, and then there were 30 patients that had febrile respiratory illnesses and were confirmed to have some other virus or bacteria. And we also had 3 common coronavirus patients that were common coronavirus positive but COVID negative. Then our positives, we had 57 COVID-19 positive samples from patients who were confirmed positive by RT-PCR at Mass General Hospital and were in the hospital mostly. We also had sort of clinical data associated with these. Next. And so for us, this is the sort of sensitivity and specificity data that we had for these 3 different assays. I will say, as a general rule, they all performed quite well. They had very high specificities. We only had 1 false positive in that cohort in this one assay. And we have tested a number of these in other negative setting positives that we know of. And then the sensitivity of these is certainly a function of time, right? And so as a patient is farther past symptom onset, our sensitivity to pick them up increases with serological assays, which is actually just the opposite of PCR testing. So as people get farther on in the infection, it's harder to pick them up by PCR testing, and it's easier to pick them up by serological testing. And so if you get to a point where you're about 10 to 14 days out, we're able to pick up most patients who've had a positive COVID diagnosis. Next slide. Thanks. And these sensitivities and specificities we have are really concordant with our in-house ELISA. So we're considering our ELISA as a gold standard as our most sensitive test. And so this is the titer of the antibody on the y-axis here. And then the x-axis here is showing this is pre-epidemic patients. So these are all negative patients, and they all showed up negative by the lateral flow here as well as the ELISA. These are COVID-19 positive patients. And you see that there are a bunch of negative patients here, and those are patients that are too early in the infection for us to pick up their antibodies. Right? And so these patients are also typically negative by the ELISA. So our ELISA and lateral flow assay data is incredibly concordant, actually. Next slide. And so this is a figure that was put together by one of our lab directors at Mass General Hospital. And this is using sort of the Wolfold data that was published in Nature a couple weeks ago, looking at PCR over time and the sensitivity of being able to detect somebody who had sort of a mild infection over time. And they were basically swabbing these patients every day. And then the IgM and IgG data here in red and blue is data from our patients at Mass General, and looking at our ability to detect these patients who we know are COVID positive as a function of time. And all of this is anchored on the days after symptom onset. And so if you know a patient is symptomatic on, say, a Saturday, how quickly would you likely be able to pick them up with the IgM or IgG assay? And this is really important for the interpretation of these results, right? And so again, the farther somebody gets out from symptom onset, the less likely you are to be able to detect them with PCR, and the more likely you are to be able to detect them with antibodies. Next. And this is really just the key takeaways. I think for both PCR and for serological testing, which is that really all the interpretations of test results should be anchored on days post-symptom onset. PCR is still and should be the diagnostic gold standard for an acute infection, right? There's the best way we know that viral titers are highest at or near symptom onset. And so we're not gonna be able to detect those with serological assays, and that's when people are most infectious. And so the gold standard here is PCR. But PCR sensitivity does decrease with days post-symptom onset, and that the serological assay sensitivity increases days post-symptom onset. And I think number 5 is very important, that a negative PCR serological results do not rule out a SARS-CoV-2 infection. There are times, especially sort of in that like 6 to 9 day window post-symptom onset, where it's possible to have a negative PCR and a negative serological result and still be positive for SARS-CoV-2. And you should never use a serological assay alone to rule somebody out for SARS-CoV-2 infection. It's very important as well. All right, and then I think next I just have an acknowledgement slide. Oh no, I think this is, as people think about how to get people back to work and policies surrounding that, this is very basic, but I think important, which is if you have the ability to do both a serology and an RNA-based antigen or PCR-based assay, that the 4 categories of patients that you would see are people who are negative for both, and those would be considered vulnerable patients. Those are people who are at risk for infection. You have people who are negative for serology but positive for RNA. Those people need to be quarantined, right? Those are people who are actively infected and are likely contagious to other people. People who are RNA-based and have a serological positive result, those people likely are still infectious and need to be quarantined, but they have potentially immune protection that's evolving. And then I think the population that we would all like to be at some point, which is SARS-CoV-2 negative, RNA-based negative, so you're not actively infected, but you have antibodies and potentially immunity to this virus. Again, we still don't know for sure if a positive antibody result means to have immunity, but yeah, we hopefully will have results on that at some time in the near future. All right, next. Yeah, so I'll just say that there's a whole lot of people that worked really hard to get all these assays up and running at Mass General Hospital, both for PCR and serology. And these are the people that were most heavily involved in the serology testing. That's all I have.
Great, that was really phenomenal. Thanks so much. I have so many questions. I was thinking that, and people are asking questions from the audience too, I'm going to ask my first question to Dr. Rosenberg, who's the head of the microbiology laboratories at Mass General Hospital. And we're so lucky at Mass General because we have so many resources in the lab, and yet I could still see how much work and effort it has been to really, shift the responsibilities of the lab to be able to do this testing. And I wonder if you could speak a bit, Eric, to kind of how the lab has had to shift and what things are not getting done and what things are getting done, and how you see that, especially in the context of Stanley and Melis talking with us about their systems in Haiti. Just your reflections.
Yeah, no, thank you, Louise, and thanks so much for organizing this, because this has been so important for all of us. And in our clinical microbiology lab, it's been a pretty interesting paradox, because on one hand, as we pivoted towards bringing on COVID or SARS-CoV-2 testing, we only had a handful of technologists who were really well trained in molecular diagnostic techniques and methods. And so even though we have a large lab, the burden of testing really fell on the shoulders of a handful of people. And what was paradoxical is, as the COVID-19 pandemic started to blossom, our other laboratory business started to slow down. And so on one hand, we had this disproportionate small number of people who were overwhelmed with testing tasks. And on the other hand, the routine stuff that we do, say rubella serologies in, just somebody who needed to go to school, that sort of business dramatically slowed down. So that was sort of reflection number one, is that there's this disproportionate task being put on people based on the skills that they have. The second reflection, and I think the one that will stick with me the greatest over the long term, is that when you have a clinical microbiology lab and you're sort of wedded to different platforms, you're only as good as the company that's supporting that platform. So for example, we have a very high throughput molecular detection system, the Roche Cobas 6800. And as Melis articulated, that is sort of our workhorse for everyday viral load testing. And that was just a giant instrument sitting all alone in a room until Roche was able to get their test up and running and provide us with reagents. Same thing with the Cepheid gene expert that we have. That's our workhorse for flu and RSV and other molecular tests. But we're only as good as the company Cepheid, and until they were able to put forward reagents, we couldn't do much. And that's why when we initially realized we have to do something, Melis and group put together an old-fashioned home lab-developed test, which was very low throughput. But those were really the biggest challenges. And it was only on the low throughput older technologies that we had the flexibility to develop in-house testing. And so we sort of used one to bridge to another, in order to get us the throughput that is now, hopefully, it's not what we need, but it's at least adequate to take care of our sickest patients.
Yeah, thanks for that. And I see Dr. Iafrate and Dr. Kerry have joined us. Hi, we'll come to you in a sec. So one of the things that I think just demonstrates, health inequities globally is that, what I see in our own hospital, we were able to start with PCR as a gold standard, work through what the serology could offer, spend time trying to understand, validate it, understand where it fits in. And I really liked, Tyler, your last graphic showing the timing. But what's happening in Haiti, and I suspect in other places, PCR is so limited in its availability, even though the technical knowledge is there and capacity. And then serologic tests, which are being wildly made by companies all over the world now and coming onto the market. Rapid diagnostics in serology are becoming really available. And that's, I think, what Stanley was telling us in Haiti. There are rapid diagnostics for serology available, but they're not coupled with PCR. So maybe I'm going to ask John, your thoughts on this, because what we've heard, I think, clearly from the pathology department is, PCR is a gold standard. Serology has a role, but you want to be very careful about the timing of when you're doing serology, what it means, and understand that. Is that the right interpretation for our audience members who are listening on how do they use these serology tests that are flooding the market?
It's a tough question, and in many ways I would defer to Eric on the right order and use of these different tests. The point of care tests, they have the advantage of they're easy and fast. They only need a little bit of blood. They can be done in 10 minutes. You can immediately triage someone, and that's incredibly valuable. We have rapid PCR assays too, but probably not as easy to deploy in Haiti or in the field somewhere. I think we've looked, including Tyler, has spent a lot of time. I don't know if you got to it already, Tyler. I'm sorry, I came late. But we've looked for antigen tests, which are using tests to detect the viral antigen as a rapid diagnostic tool. Those aren't quite ready yet. We had them very early, but there's some hope that they will work. They won't be as sensitive, and Eric can confirm the importance of sensitivity in this disease. They're not going to be as sensitive in our opinion. I think all of us have expertise, certainly expertise, but some experience looking at the data. And if it's not sensitive as PCR, certainly in a place like Boston, there's very little use for them. If there's no reason that we would want to replace something that we know is very, very sensitive. So in many ways, working as hard as you can to get a PCR assay up and at scale makes the most sense. But again, I would defer to Eric as the expert on that topic. From an antibody detection serology standpoint, yeah, they're easy and fast, and they can be used to triage people really, really quickly. We've attempted and been successful in using these in our own communities just this week, and we know that they're robust and easy to use. Some of them will go through EUA approval in the US, some of the manufacturers, and I think some of the very large manufacturers, we're working with a company, Becton Dickinson, which is a very famous worldwide medical supply, medical device manufacturer. And so having companies like BD behind them and manufacturing them changes everything, because I think their stamp of quality will make a big difference in especially in the third world, because you don't just want just because something's inexpensive which is attractive, is not going to make it quality or help the local community. So if we can get either EUA approval or some of these manufacturers to distribute globally, and they're scaling up as we speak. So I think I think that's a very hopeful sign. Now, Tyler probably described that in the first 5 days, it's not going to be useful. And so during the acute infection process, they're just going to be negative. So is there value in Haiti or in other resource limited countries to saying in the first 5 days, we can't tell you, come back in a week? I don't know if that's of value, but I will tell you there's likely going to be value for social distancing and identifying individuals who have been exposed and possibly are shedding, and coming up with some common sense rules about where to direct those individuals or how they should behave. And if that can slow down even a little bit, I think it's worth considering and possibly deploying. Likely, I'm not epidemiologist. Our team has not focused on that, but I think as the folks in Boston begin to chew on the data that's being created, some models could be made about if you had this test at day 7 and you use that information to inform behavior, social distancing, and other behaviors, could you bend or flatten the curve in those communities? I think it's definitely worth trying, but again.
So I think the short story that I'm hearing is the rapid antibody tests are good at finding the antibody when the antibody's there. Like, they're good at detecting the antibody. The issue is that we must use them understanding the timeline of when antibodies are formed. Absolutely. And making sure that the use of them is thoughtful in the context of understanding what it's telling you. So I think that, you know, for folks who are taking care of patients with a respiratory illness at a clinic in, you know, rural Tanzania or rural Haiti, they need to be very careful about what that antibody test result is actually saying to understand it. I think that's
the graph that Tyler showed is, it's critical for us. I mean, Eric and Jill Lenners in our group spent a lot of time with Tyler and Melis also figuring some of that stuff out. That was just last week, right? Where I think the thought process about these diagnostics just was changing on the fly. It's not like something we've known for 2 years. We've just been working on this for a few weeks. So it's really impressive what they put together. I think it's as important here as it will be in Haiti.
Yeah. So let me ask one more big picture question to Dr. Kerry, who studies health systems, and then we'll come back to some of the specific technical questions that folks have about Haiti and about some of the tests. So Vanessa, I'd love to get your reflections here. One of the things that Stanley, Juin, and Melis, Pierre in Haiti were telling us early on is that, you know, they need reagents, they need equipment, and in the global marketplace right now, it's almost impossible to compete for purchasing PPE or reagents. Can you just give a little reflection on that?
It's funny, I was going to actually text you to say, there's so many interesting questions. Don't worry about me as a discussant, like just go to the questions because I have so many too. But I think that this is a very important point. Obviously, the testing in less resource-rich settings is complicated, one, by obviously the complexity of getting reagent swabs and all these things that might be needed for PCR, which really means that probably the fastest, most effective in terms of scaling a test up is going to be the antibody test. And we've just heard an articulation of why that's a difficult test in terms of preventing ongoing infection, because obviously what we'd love to do is to capture people when they are at the height of infection, have the highest viral loads, and can cause the most problem. And I think that as a result, the issue actually Bill and Melinda Gates have put in a huge amount of money to be able to get testing done that could be done at home in 10 minutes without electricity, $1 per thing, which is this antibody test. And the question really is, how do you create not just the testing at scale, but how do you create the mechanisms to support an entire system? Because what happens when somebody does test positive? What is the significance of that? How do you quarantine them? How do you make sure, one, you can get the test to every household in an effective way? How do we ensure that we know what to do with that information when we have it and can support individuals, communities, and households with that information? And make sure that if we think they do need to quarantine based on overlaying that result with a grid of where they are maybe in an infection cycle or how symptomatic they are, and knowing that up to 80% of patients with very mild disease are asymptomatic, it's going to be very difficult to, I think, create a clinical diagnosis or a clinical sort of decision-making tree that overlays a positive test result. And so I would agree, PCR is going to be would be the most effective in terms of shutting down infection. It's a real issue about the ability to create a market to buy these reagents and things that would be needed to be able to get a PCR or that kind of sensitive testing to scale. And I think the best bet is going to be, and there is discussions about doing this already, having an African Union-like approach to create a market with the 1.3 billion people and the 54 countries that make up an African continent. And I'm not trying to exclude other countries that maybe aren't on the continent, just that my work is most based there, so I can understand creating that market a little bit more. That's what was done for HIV medications back in 2005, 2006, was a group market was created for all the resource limited countries that brought the price of antiretrovirals down significantly. I think you could create that kind of purchasing power through some sort of global fund, again, to create the markets. COVID has shown time and time again over the last 2 months that no one is immune. Everybody's at risk, and this is a global pandemic issue. And so there's a real incentive actually to create a network that protects all countries and creates a system for everybody to have access to testing. I think the final thing I would just add is that what is very real also is kind of managing stigma around this. Stigma is a real issue in terms of also rolling out testing, because much as we saw also with the early days of HIV, when you have the disease, I think that people are often getting discriminated against, and this has been called in some countries in Africa a white man's disease already. There's already been backlash and violence against healthcare providers who are seen as potentially higher risk of getting the infection or having or bringing the infection if you happen to be an Asian, of Asian descent or of a non-African descent. And so dealing with stigma and some of those pieces too is going to be important. So I know it's a little bit broader than just talking about the purchasing power question, but I think there's a lot of issues that are going to come into the question of rolling out testing. And of course, near and dear to my heart is the healthcare providers needed to manage the results when you see it and the triage system. So as I say that and lay out an overwhelming list of problems, all of those are surmountable and all of those are solvable. It's just about being creative and creating the community needed and the conversations needed to begin to sort through those in a very fast and real way.
Yeah, thanks. A test is never just a test. It's the whole system around the test. I think let me go back down. Thanks, Dr. Kerry. Let me go back down to Haiti or up in the corner of my screen, because we have a lot of questions. I think probably folks from Haiti on the line asking some things like you mentioned about GeneXpert. So we're used to using GeneXpert in Haiti, but we usually have much smaller machines than what we have in Boston. I think probably 4 cartridge or such. Can you talk a little bit more about that, Stanley? And also I know that the Cepheid test cartridges were quite expensive, I thought. But can you tell us a little bit, are you going to roll that out? Will it be centralized? Will it be decentralized? We'd love to hear a bit more.
Okay, so Melis wants to say something, and I think I can talk about after Melis.
Because the plan is to decentralize with the GeneXpert, the fact that they've been using it for TV throughout the country. Like Stanley mentioned, the total 40, but obviously we're not going to deploy the cartridges to do COVID tests to all 40 locations. However, we have a plan to have them at key central places in the north, south, but we have the first problem is limited resources as far as PPE we mentioned before. But also I wanted to know about the type of specimen that they use to do validation, because we've realized doing certain validation that the sample, collecting a good amount of sample is important, as well as the assay that we're using. So that's one of the reasons we wanted to limit the laboratories will be doing COVID testing for that reason, because you're familiar with Haiti, finding a lot of already trained staff for a new technology can be difficult. I wanted to know more. They will focus on the staff who's been doing TV tests, but we wanted to know if there's similarities, also what we need to know, what your experience has been if we want to go that route.
Okay, good. Mm-hmm. Also, in terms of it's quite expensive, the GeneXpert testing. There have been some effort from the private sector here in Haiti. I think it was led mostly by Bill Papp and Dr. Bansi, and they requested from those guys a lot of GeneXpert tests. I think the plan from is to have about 17,000 tests and to start from there. So the first batch will be procured by the private sector, but CDC on our budget, we included some fund to buy GeneXpert as well, and we also plan to buy 1, 2.
Oh, I lost Stanley. Oh,
you lost me?
We heard you saying you were going to buy 1 or 2 something.
2 equipment.
2 equipment.
Okay.
So
for GeneXpert. So I think what we can do, I know that our colleagues here on the line have proposed to offer some technical support around validation, and we can certainly do that separately. So I'll follow up with you on that, because we just have a few minutes left in this call, but I know that we can do that. A couple of people are still asking questions, which I think I'll turn to Tyler for. So you mentioned in your presentation that you've looked at some of the rapid antigen tests. Can you just say a little bit more? They don't seem to be performing well, or because there's a lot of interest in would that be a useful tool?
Yeah, I think for a low resource setting, it makes a lot of sense, even in a high resource setting, to be able to screen in the community at workplaces and stuff, to have a rapid antigen test that's available. I think our issues so far that we've tested, they haven't had great sensitivity. But if you could identify even the highest titer individuals, and those are likely the most infectious, it might be of use. I think that the other thing that's an issue with the rapid antigen tests that we've tested, they've all required nasal swabs. And so there's obviously issues with supply of nasal swabs and viral transport media that these use, as well as the fact that you should really be using PPE in those scenarios where people are testing with nasal swabs. So I think an ideal situation would be to have a rapid antigen test that theoretically could be used on saliva or some other sample type that was easier to acquire and didn't require any special equipment. It could be widely deployed. And even if it had sensitivity of 60 or 70%, then that still has potential utility, especially in lower resource settings.
And I don't know who from you, maybe Melis could take this. So related to that question is some reflection on whether the alternative methods, like what about 3D printed swabs, and how about sputum? Like, are there other approaches that we could try to use, especially as we're trying to bridge the gap until the Cepheid cartridges are more widely available? Because even at MGH, we've been limited in how many of those cartridges we could get, right?
Yeah, we've certainly explored different transport medias. In fact, we're now using this prime store molecular transport media, which has an advantage of inactivating the virus at the time of placing the swab inside of the media. We've looked at saline and then different additives to saline using phosphate buffered saline with albumin and whatnot. So there are certainly many options, and there's a nice paper published by the Mayo in the Journal of Clinical Microbiology that shows that many of these transport medias are equivalent in their hands, though we've seen some degradation in saline over time that is different from that data, but we can talk about that offline if anyone's interested. In terms of swabs, there are people in Boston and elsewhere who are working on the 3D printing, and they're clinical pathologists who feel that the quality of those swabs is equivalent to the flock swabs that we normally use. We haven't personally used those 3D printed swabs, but that is an option that's now looks like it's becoming more and more available. But yeah, the reagent supply issues in general is a never-ending problem that we have had to deal with. I don't know if Eric wants to comment any further.
No, I think you addressed those issues completely appropriately. The one thing I might add is that we do need to get creative about strategies, and whatever resources available, there may be ways of optimizing how that resource is used. So for example, if you have antigen testing available, even if the sensitivity is only 50 or 60%, it may be something that linked to a very rapid point of care antibody test, you may be able to really crank up this sensitivity and the specificity. Mm-hmm. It's not going to get you close to PCR, but I think in these new days, it's just we have to make the best of what we have and try to sort through the most creative and instructive strategies. So I would put whatever you have, I think we should just put on the table and try to figure out ways of optimizing that as a first move.
I think that's a really great comment, Eric, thanks. And maybe I'm going to relate it to that. I'm going to give the last word to Stanley and Melis, because you mentioned earlier that there was resistance to using rapid diagnostics And when I say rapid diagnostics right now, I mean antibody testing in Haiti by the Ministry of Health. But wouldn't it be a good idea to use that as one tool, just knowing what the tool is telling you? You know, so you don't want to use it for an asymptomatic person to diagnose them, but you know, just understanding maybe in surveillance or community surveillance. Could you comment on whether there's openness?
Before he goes,
Stanley, could you comment if there's openness to that approach in Haiti, and then I'll come back to you, John.
Okay.
I think so.
As of now, there are discussions, but mainly within the ELR for the use of those antibodies rapid testing. And how we see them using it is like, since you know we have very low positivity rate up to now, but since we are also seeing some positive that we cannot link to any case that we know of, so there's a need to go to the community and at least do some screening within a specific community to see whether COVID has been circulating in the area. Then we could focus on this community to do contact, to do PCR first to confirm, and then we could quickly identify those cases and isolate them and quarantine those that needed to be quarantined. There is openness to this. But I also think we could use it as Eric said, to quick use for symptomatic patients, and we could do this. But there's a fear here that once we open to that, we will have private labs doing COVID testing and giving positive and negative results to patients. And this is a fear from of the Ministry of Health. So they feel like if they open this, it will be a can of worms where it will be a free fall. Everybody will be testing, and people will say that they are negative. And we actually saw like in one instance, one case that it was positive, and then they follow up after because it wasn't symptomatic. So here the standard is to test them twice within a span of 24 hours before releasing them from isolation or from quarantine. And when MSPP called him, he said, "Oh, I was tested at another lab, and they said I'm negative, so I don't need to come back for the test." And these are some issues that we fear may happen. Right. So that's why MSPP is dealing with this with. Right. Yeah, they want to deal with this with. They want to be precise when they make a decision. And for now, I think it's only MSPP would be allowed to use it and in specific settings.
I understand. We are over time, so I'm sorry, John, not to come back to you, but I think we're probably going to have another conversation about this in the weeks to come. I really appreciate all the panelists. I know every single person is super, super busy. We really appreciate your expertise, appreciate you calling in from Haiti, Stanley and Melis. And thanks to everybody. We're going to post this video on our new YouTube channel and we'll share around the link. Thanks for joining us, everyone. Have a good day. Bye.
Thank you.